Alright folks, let's talk about chapter 5, which is going to be about the microbial world. So this is basically anything that is microscopic. So here's just a simple breakdown of the major groups within the microbial world. And you can see for organisms, it's split into two categories. Organisms which are considered living, and infectious agents, which at the moment your textbook considers non-living. And we will get into, when we talked about in chapter 4, I believe it was, the fundamentals of life. That's where we talked about what constitutes living versus non-living. And what we talked about was that viruses, since they don't have, carbon, hydrogen, and oxygen, those three main elements that contribute to carbohydrates, proteins, nucleic acids, and lipids, because viruses don't need to take advantage of or don't have those elements incorporated into their cells. Amongst other things, they were not considered living, and everything else is considered living that does take advantage of those three main elements. So you see the two categories. You have your living, your non-living. In your non-living, you have viruses, viroids, and prions. So those are just different types of viruses, basically. And then on the living side, we have our three domains, bacteria, archaea, and eukarya. And all three of these domains have microscopic organisms found within them. In the bacteria and archaea domains, you can see we have prokaryotes, which are single-celled organisms that are very simple in nature. And then you have your eukaryotes in eukarya, which are more complex and have membrane-bound organelles. And so we'll talk a little bit about that later. And then the eukaryotes are further separated into whether they are algae, protozoa, fungi, and whatever that last one is, multicellular parasites. I'm not even going to bother trying to pronounce that word. And then the algae and the protists are further separated into protists, which used to be its own kingdom, but is no longer a kingdom because they noticed that so many of those organisms that they previously classified as protists actually were, say, bacteria or archaea. So they were classified wrong initially. Okay. So let's talk about viruses first, since they're a little bit more simple. But viruses are not made up of cells, as you can see in the image on the right-hand side there. They are made up of two main parts: the caspid, which is that head part, and the genetic material within the caspid. And then the other major features of viruses is, generally speaking, depending on which one you're talking about, the one on the left-hand side has a head, a tail, and a base plate where the DNA is injected through the tail, out the base plate, and into the bacteria. And we'll cover this in a little bit. But some other viruses are the ball-shaped ones that are on the right-hand side there, and the DNA is on the inside, and it has the outer covering, which is the caspid, and then it has those appendages, which are neuraminidase, basically just connectors that will how do you say this now? It's really late. My brain's a little tired. Anyways, those connectors on the outside of that round... Be quiet. I'm talking. No, be quiet. Those receptors on the outside of that round virus there will connect to the receptors on the outside of bacteria. and they will trick the bacteria into thinking they're a friendly cell, but really they're just there to attack them and take over the bacteria. We will see this in a little bit. But essentially, viruses can contain either DNA or RNA, just depends on what type of virus they are. The caspid is a protein covering that protects them, but they don't have any lipids, they don't have any carbohydrates, so they do lack some of the basic things that living cells have. And viruses cannot replicate on their own. They have to infect a living cell in order to replicate and reproduce themselves. So they don't do any sexual reproduction. They don't do any genetic recombination of the chromosomes. Their DNA is just that. All right, so let's take a look at this. There's a couple of different ways. In particular, there are these viruses known as retroviruses. These particular retroviruses contain RNA instead of DNA, so they only have a single strand of RNA within their caspid, and they typically are associated with some of the more deadlier human diseases like AIDS and leukemia. And the image that you see there, you can see how a retrovirus replicates. This is pretty common for most of the viruses, but in particular, retroviruses are known for doing what's called lytic virus replication. So in a lytic replication cycle, the virus attaches itself to the outside of the bacteria cell, which then the virus proceeds to inject its RNA into the bacteria cell. So it penetrates the cell wall of the bacteria, injects its RNA from the caspid through the tail and through the base plate into the bacteria cell, whereby the RNA that has just been injected takes over the bacteria cell, and the bacteria cell then no longer functions as a regular bacteria. All the purpose of that bacteria cell now is to replicate all the needed parts to reassemble more viruses. All right, so you can see in part C there, it's starting to replicate more DNA, and this is not its own DNA, this is the virus's DNA. It no longer replicates its own DNA, it no longer eats or photosynthesizes. Whatever it was doing before it got infected, that's it. It is like a robot now, in a sense. And then in part D, you can see that it's starting to produce the caspid, the head, the tail, all of the needed parts to put back together this virus. And you can see it's producing as many copies as it can make. And remember, it's not eating anymore. It's not gaining any more energy. It's using up whatever energy, whatever nutrients, whatever materials it has within that cell in order to reproduce this virus. So when you look at part E now, you can see that the virus is starting to become assembled. So again, this bacteria isn't doing what it's supposed to do. All it's doing is reproducing this retrovirus. So it's starting to assemble the individual viruses. And once all of the individual viruses within that bacteria cell have been assembled, it moves on to part F, where those now fully matured viruses break open that bacteria cell and are released back into the environment. Now, that cell, that bacteria cell that it left behind, is dead, basically. Once it has ruptured that cell wall, which is another way of saying lytic. In the science realm, when you say lytic, it means that something has ruptured. And so those viruses have ruptured that cell and caused that cell to die. Not only for the fact that it ruptured the cell wall, but also it used up everything within this bacteria to replicate themselves. So that cell no longer exists anymore. It died. You could probably say it died when the virus injected its RNA into it, but that's a more philosophical question. But yeah, so that is what's known as the lytic virus replication. But there's two different types of replication that viruses can do, and the more destructive one is the lytic cycle. But not all viruses immediately do the lytic cycle. Some viruses will actually do what's known as lysogenic replication, which this is more common. This is more of what AIDS does, so HIV and AIDS, right, go hand in hand. So HIV does the lysogenic replication, and once HIV turns into AIDS then does a lytic replication cycle. So when somebody gets infected with HIV, that virus then injects its DNA into the cell, where you can see, let's see. Okay, yeah, so at the top there where it says lytic, and then it says attached to host and inject DNA. So the virus will then come in, insert its DNA into the cell, but once it goes down into the middle there, and you see that red circle and the black circle represents the bacteria's DNA, the red circle represents the virus's DNA. And when HIV infects a person, it goes to that step in the middle there, and then it follows the green arrow to the right, where the viral DNA integrates into the host bacteria's chromosomes. So it incorporates itself into the black circle. It does not follow the purple arrow to the left there. And so then when HIV gets into your body, it continues to spread using this lysogenic cycle. So it'll keep infecting more and more cells, because those cells will continue to replicate, and they will replicate that virus DNA along with their own. So they function as regular bacteria or regular cells, and they don't know that they were injected with the viral DNA until it gets to a point at the top there where it says, when triggered, the virus becomes active. And in the case of HIV, they're not quite sure what triggers HIV. A lot of these viruses that do the lysogenic replication cycle, there's really not much known about what triggers them, but at some point something triggers them, and it causes that viral DNA to separate itself from the bacterial DNA. As you can see there, that red circle is starting to pull away. It's no longer being incorporated, and once it pulls away, it goes back into the middle, where then it proceeds to take the left-hand fork and follow the purple arrows and start the lytic replication cycle. So this is where it goes into overdrive and starts making more copies of itself, instead of just copying the DNA in the lysogenic cycle. So on the left-hand side, the green cycle would be your HIV. The purple cycle would be AIDS once it converts into that. All right, so then there's these things known as bacteriophages. Bacteriophages are viruses. They look just like what's depicted in the images down below. That first grainy looking image is what's known as a scanning electron microscope image. And that image is able to take a picture of a virus which under normal microscopes you wouldn't be able to see. and you can see the clearly the head and the tail and the tail fibers. All right, but these particular viruses don't really attack humans like HIV and AIDS do, but these particular one attack bacteria. Hence the reason why they're called bacteriophages. They attack and destroy bacteria. So their whole purpose is just to destroy bacteria. And they are super common in the ocean, and they play a very important role in the ocean in terms of, how do you call it? Returning the nutrients back into the cycle of things. So when a bacteria dies, that material that makes up the bacteria goes back into the ocean cycle, basically. So that bacteria then falls down to the bottom of the ocean, and when it gets down there, it's then decomposed, and when it decomposes, those nutrients that make up the bacteria are then recycled back into the ocean, which then those nutrients come back up to the surface and are taken advantage of by other bacteria, right? So it's a whole cycle of life, and they are common and also very important in that role. And this particular virus, the abundance of this virus is determined by how much bacteria is within the ocean, obviously. Because if there isn't a lot of bacteria, then you don't see a lot of bacteriophages, because that's the only way that these particular viruses can reproduce. So when you go into the ocean, you're probably surrounded by a million of these guys in one small little area. But you don't have anything to worry about because they only attack bacteria. They would not attack your skin or your cells. If you had bacteria on your skin, they would probably attack the bacteria, but not you. So don't worry, don't freak out the next time you go to the ocean. It's fine. All right, so this is what I was talking about on the last slide. Viruses contribute to the overall dissolved organic matter in the ocean, and this is essentially the cycle of nutrients within the ocean. And the cycle of nutrients basically starts off with your zooplankton and your phytoplankton up at the surface, and once those die, they contribute to the overall dissolved organic carbon, dissolved organic nitrogen, and dissolved organic phosphorus. Those are DOC, DON, and DOP. And those are nutrients that are dissolved into the water column, and the bacteria, those heterotrophic bacteria, are taking advantage of those dissolved inorganic nutrients. All right, so because they need nutrients to grow. And then once those bacteria grow, they contribute to the virus population, because those bacteriophages then will attack those bacteria and break them apart and allow them to contribute back into the dissolved organic nutrient pool. Okay, so it's all just a cycle. Those heterotrophic bacteria also create inorganic nutrients, which then are taken up by phytoplankton and algae and used to create oxygen. So some of these viruses, though, can infect organisms such as shellfish, sea turtles, marine mammals, and humans, but typically humans don't really get affected by viruses that you see in the ocean, unless, of course, you eat shellfish, right? So be careful what you eat from the ocean. Make sure you cook it thoroughly, and if you don't cook it, make sure you know exactly where it came from, because you definitely don't want any contamination. But normally speaking, when you go to the ocean, your body more or less takes care of all of the bacteria and viruses that are in the water so they don't infect you. But I wouldn't go gulping down huge amounts of seawater, aside from the fact that it's not good for you because of the salt concentration, but also because there's a lot of viruses and bacteria in the water. All right, so let's talk about prokaryotic cells. I know we talked about it in last chapter, but this is a little bit more in depth in terms of what is what parts of the prokaryotic cell there are. So on the very outside, that yellow cell wall thing is what's known as a capsule. So this is a very hard protein like not really shell, but outer covering that protects the bacteria. And usually on the capsule, you'll see pili, which are these thin hair-like structures. Pili are not generally used for locomotion. That's usually reserved for cilia or the flagella. Pili are generally used for sensory. They can sense things like vibrations and whatnot. And then the green covering that's under the yellow capsule is your cell wall. So this is the generally made from carbohydrates. And this is just an additional layer of protection for the cell and the genetic material on the inside. The white covering underneath the cell wall is your cytoplasmic membrane or the cell membrane, and that's just an additional layer of protection. So in this particular case, you can have as many as 3 layers of protection, but not all prokaryotic cells have all 3 of them. Some may only have a cell membrane, some may have a cell membrane and a cell wall, some of them have all 3. It just depends on the specific type. And then within the cell membrane, you have the cytoplasm, which is a light blue color, and that's basically just the fluid within the cell that keeps the genetic material within it. And then you tend to have ribosomes, which are the small little, I guess they're greenish dots within the cytoplasm, and they produce proteins for the cell. And then you have your flagella on the outside, and that's mainly used for locomotion. And then you also have your nucleoid, which is just a fancy word for the nucleus. And that's generally where the genetic material is stored. Now keep in mind this genetic material, which is typically DNA, is just floating in the cytoplasm. There's nothing surrounding it or holding it in place. So it pretty much just goes wherever it wants to within the cell. Typically, you'll see prokaryotic cells as oblong shapes, but you can find all different shapes and sizes for these prokaryotic cells. And you can find these particular guys within the bacteria and archaea domains. You do not find them within the eukarya domain because those are more complex cells. So for bacteria cells, you can see they vary in a lot of different shapes and sizes. Typically, you'll see the oblong shaped ones, but you can see the coccus, which are just small little round balls. You can find rods, which is the oblong shaped one, or bacillus. You can see spiral shaped ones, or let's see, what else is there? Spore forming rods. Well, that's really interesting. Or you can get cocci in chains, which is just the small little round balls all grouped together in one long chain. You can get the round balls within a square shaped or cube shaped. So you can pretty much find all sorts of different shapes and sizes. They can come in rings, rods, spirals, spheres. any shape you want to, really. These are just some examples of them. And then you can have different functions for bacteria. So much like how there was bacteriophages for viruses, bacteria also have what's known as decay bacteria, and these particular bacteria are responsible for breaking down any organic material that that is found within the ocean. So whenever a plankton dies, it turns into a waste product, and that bacteria then goes and decomposes that plankton's body into nutrients, which then are released back into the water. And these particular bacteria are also responsible for creating oil, basically. So you have decaying organic material that gets trapped at the bottom of the ocean, and then it's covered by sediment. And over time, that degraded organic material then turns into oil over a really long period of time, mind you. But eventually it will. And then some of these decay bacteria are responsible for diseases. So a lot of the shellfish diseases are because of decay bacteria and spoiling of food. So if you guys remember the outbreak, I believe it was hepatitis A outbreak for the scallops at Genki Sushi. Well, the reason why that happened was because those scallops not only had hepatitis A, but they were also going bad. So definitely be careful where your seafood comes from, and make sure you are not eating bad seafood, because you can get very sick from eating spoiled seafood. All right. So another type of bacteria that you can see are known as cyanobacteria. Back when I was younger, I knew this category as blue-green algae. So they thought this type of bacteria was actually an algae, but in reality, they discovered that it was actually a bacteria, and it was just long chains of single-celled bacteria. So you can see in the image on the left-hand side there, you have round balls all connected together, and each round ball is a individual bacteria. And so you can find long strings of these bacteria all attached together. And these particular bacteria are photosynthetic, so they create their own energy, so they don't infect other organisms like the decay bacteria does. And these guys were thought to be the first photosynthetic organism on Earth, and were thought to have a large contribution to oxygenating the Earth's atmosphere. So those particular what look like rocks on the right side image there are known as stromatolites, and stromatolites are just bundles of cyanobacteria, and they produce a ton of oxygen. And so these guys are thought to be the oldest living organism on Earth. And we can find fossils of these guys dating back to millions of years ago. Okay. Oh, you can find those stromatolites in Australia, by the way. They're still alive. They're still there. You have some bacteria that are known as endolithic, meaning that they burrow into the calcareous rocks and corals. So these types of bacteria will live under the surface of rocks and corals. So if you look at the left-hand image there, you can see the layer of black and green on that rock. That black layer is bacteria, fossilized bacteria. And then on the right-hand image there, you can see that reddish hue in the ocean. Those are created from bacteria. It's what's known as red tides. So you can have dinoflagellates create red tides, but you can also have bacteria create red tides as well too. And then you have others that are known as epiphytes. Epiphytes live on top of algae, so they live on the surface of the leaf of the algae. But then you can also have endophytes, which live inside of the algae. So much like the endolithic bacteria that live in the rock, the endophytes are bacteria that live within the leaf of the algae. Next, we have archaea bacteria. These are known to be the most primitive life forms because they're only prokaryotic bacteria. And these particular guys are thought to have played a very important role in early evolution of life because these particular bacteria are also known as extremophiles. So we'll get into that on the next slide. But there is some evidence that suggests that these bacteria in the archaea domain are very closely related to eukarya. So the bacteria that are within the eukarya domain. And you can find all sorts of shapes within this domain as well too. You'll see spheres, you'll see spirals, you'll see rods, and any sort of combination of those as well. So the reason why they're known as extremophiles is because all of these bacteria can be found within very extreme locations. So for instance, you'll find these types of bacteria within hydrothermal vents where it's really, really hot, but they do just fine. They'll be in geysers and hot springs where generally other organisms will not be found in. And you also find them within the Dead Sea or in a desert. Or you can find them in a glacier too. So they are in very interesting places where other organisms normally would not be found. And modern genetic testing has shown that bacteria within the archaea domain are very common within the ocean. And so not all of them live within these extreme environments. A lot of them live just within the ocean water itself. But the ones that do live in extreme environments, they will definitely be from this domain. And so the reason why these particular bacteria are thought to have played an important role within the early evolution of life is because in early Earth, before life even existed, it was a very extreme environment. You had a lot of carbon dioxide in the atmosphere. The oceans were very, very acidic. You can imagine right now the ocean pH is about 8.1, but back in that day it probably was closer to 3. It had a lot of hydrochloric acid, a lot of carbon dioxide within the water, and that was all because of volcanoes. And so it was very, very acidic to the point where you wouldn't even want to go swimming in this water. And even back then, there was bacteria within the water, and it was thought that it was archaea bacteria that lived in that water. So from that, that's why they think archaea bacteria had a very important role, because they were probably the very first bacteria found in the ocean. That was it for archaea. But there's some different types of metabolism methods that bacteria can have, and these are the three methods that they can have. You can be an autotroph, you can be a heterotroph, or you can be a nitrogen fixer. And so let's cover each one individually. We should be all familiar with autotrophs. Autotrophs are organisms that can create their own food. And so they typically will do either photosynthesis or chemosynthesis depending on where they're located at. If they're located near the surface, they will do photosynthesis. If they are located in an area that does not have sunlight, they will do chemosynthesis and use hydrogen sulfide instead of sunlight, basically, to do their chemical reactions. Next is heterotrophs. Heterotrophs, unlike autotrophs, cannot produce their own energy, so they need to eat their energy. So a good example are humans. Humans are heterotrophic. We need to consume our food in order to get our energy. But in bacteria, you have two modes of respiration for heterotrophic bacteria. You have aerobic bacteria and you have anaerobic bacteria. So aerobic bacteria need to use oxygen in order to do their respiration, basically. So humans is an example of being an aerobic heterotroph, basically, because we need oxygen in order to respire. But there are some bacteria in the ocean that do not require oxygen when they respire, and that would typically, you can typically think of it in terms of bacteria that you find when you're making wine or when you're fermenting something. So when you basically ferment wine, when you age wine, you're introducing bacteria into a oxygen-free area where it then takes the sugars, the glucose, and it breaks it down into carbon dioxide and alcohol. So that's the reason why when you ferment grapes, you get wine. The other route is making lactic acid. So when your muscles, when you go to the gym and you do a really good workout and you're proud of yourself and you walk out of that gym and you're all sore and you're like, yes, that's a really good feeling. Well, that feeling, that sore feeling of in your muscles that you feel when you come from the gym, that is a buildup of lactic acid within your muscles. And that buildup of lactic acid is basically your muscles using glucose to respire, to work, because you're putting that muscle to work. So it needs energy now. So it's using up the glucose and it's converting it into lactic acid. And when that lactic acid builds up within your muscles, it causes that soreness in your muscle. So don't worry about it. It goes away eventually, right? You don't feel that soreness after the next day. It's because your body reincorporates the lactic acid back into your body. But that is a form of anaerobic respiration. All right. Then you have nitrogen fixation. Some bacteria within the ocean are what's known as nitrogen fixers. And so they will take nitrogen from the atmosphere, and when it diffuses into the ocean, they will convert it into ammonia, which is NH4, and then it will go through, that's what's known as nitrogen fixation. And then they will do what's known as nitrification, and they will basically oxidize that ammonia, that NH4. They will turn it into NO2, which is nitrate. Yeah, nitrate. And then they will further do one more nitrification of nitrate and turn it into nitrite, which is NO3. And then it starts all over again and it goes into what's known as denitrification. and then it goes back into the atmosphere. But it can also go down and do consumption and be consumed by algae and other organisms that require nutrients. And once the algae dies or gets eaten, then it goes down to the bottom of the ocean, where then it gets decomposed and goes back up and turns into ammonia. So you have this whole cycle again. Different types of prokaryotes that you can see. Prokaryotes are unicellular algae, or protozoans, or fungi. So they can be any of the three. For unicellular algae, they can be diatoms, dinoflagellates, or just microscopic algae, but those are the main type. Or you can have protozoans, which are heterotrophic. So your algae is going to be autotrophic. Your protozoans are going to be heterotrophic, so they need to eat. So they typically will eat the algae. And they typically will be your foraminiferas and your radiolarians. So that image with the black background on the right hand side on the top, those are different images of radiolarians. And on the bottom there you can see some examples of the different types. So you have cyanobacteria, diatoms, dinoflagellates, green algae, and coccolithophores. And then also fungi. You can have prokaryotes that are fungus related. So your unicellular algae, much like plankton is right there, tend to be photosynthetic, and they can be eukaryotes, but they don't have to be. So they can be prokaryotic or eukaryotic, either one, but they will almost always be photosynthetic. So you'll find them near the surface of the ocean. And initially, scientists thought that algae were plants, but now we know them to be, I don't know if it would be quite right to say animals, but they're definitely not plants in the ocean. Some of these algae species do freely swim, so they do have a flagella that lets them swim about. Again, at the mercy of a current. And there are a few species that actually do consume food, so they are heterotrophic, but majority of them are autotrophic and produce their own food. A lot of these species in this group right here used to be categorized in the Protista kingdom, but the organisms that are found in the Protista kingdom do lack specialized plant structures. So they're not your typical plants. And Protista kingdom can have animal, plant, and fungi species all within the same kingdom. So that's the reason why this particular kingdom, a lot of the organisms within this kingdom were misclassified initially, but there has been a lot of work to date to try to correctly classify those organisms. So diatoms are unicellular algae. They do aggregate in chains or star-shaped groups. Their shell or test is made out of silica, and they tend to have a yellowish brown pigment or hue to their shell because of the carotenoids pigments that they have within them. And these particular guys do not have spines, so they use oil droplets within them to prevent sinking. Typically, these guys don't have spines. Typically, they look like petri dishes. If you can see there in that cross section, you have a top cover that sits over the bottom cover, and overlaps in the middle. And so over there you can see that the nucleus is in the middle. This entire structure is known as a frustule. So the top frustule, the upper frustule, is known as the epitheca. The lower frustule is known as the hypotheca. And then you have chloroplasts that photosynthesize, and you have mitochondria, and you have that oil drop that keeps them afloat. So those are your different parts to the diatom. And diatoms have a very interesting reproductive cycle. Typically speaking, they will undergo asexual reproduction, whereby they will replicate the material on the inside, and they will separate the two halves of their shell or their test. And the top half that you can see up there creates a new bottom, and then the bottom half then creates a new top half, right? So you can see that the bottom one of the two is smaller than the first one, right? And then you can see it replicates again. Each one of those two replicates again, and you get 4 new individuals, or well, I shouldn't say new individuals. They are all the same individual. They're all clones of each other, but there's 4 new ones now. And you can see they're all different sizes. The top one is the largest, while the second one and the fourth one look about the same size, but actually the second one is a little bit smaller than the fourth one. And then the third guy is the tiniest of them all. Okay. So the first one, the one on the top that's the largest, that guy will continue to replicate just fine. But the third one, that's the smallest one, cannot replicate, cannot do asexual reproduction anymore because it just doesn't have enough material to do asexual reproduction. So what it does is it goes through what's known as sexual reproduction. So it goes down to the bottom there, and it turns into either an egg or a sperm, and then gets fertilized and turns into an oxospore. And then that oxospore grows up, basically, and turns into a larger adult diatom. So that's how they go through sexual reproduction. It's generally those small individuals that do that. Otherwise, the larger ones will just continue to do asexual reproduction. And diatoms are interesting because they can be harmful to humans. They do produce what's known as domonic acid, which can cause somebody to feel really, really sick. Very similar to like stomach, food poisoning. Yeah, very similar symptoms to that. And when you have very favorable conditions, you can have blooms. So when I say favorable conditions, that means that there's a lot of nutrients, and then when there's a lot of nutrients, they can reproduce faster and create more clones of themselves. So you'll get a rapid replication of themselves, and you'll get thousands of these guys. So if you look at the image on the right hand corner there, you can see that brownish hue to the sand. This is an aquarium tank now, and that brownish hue in the sand is a bloom of diatoms, right? And when you have large amounts of diatom tests, or their shells, that fall to the bottom of the ocean, they create what's known as a diatomaceous ooze. and or siliceous ooze. And this material is basically just their shells. And this material is made out of silica, remember, and so it's really good at filtering out pool water. So it filters out any bacteria or viruses or debris that is found within pools. You can use it for temperature and sound insulators, and you can use it as a mild adhesive in toothpaste. But diatomaceous earth is also used in pest control as well too. So if you have those nasty little ants outside, you can use diatomaceous earth, circle around your house with it, and that will get rid of them. Next we have dinoflagellates. These are also unicellular, so that's only a single cell. They are planktonic, so they're microscopic, but these particular guys are eukaryotes, so they are more complex, right? Typically, they tend to have two flagella, and one will be wrapped around the center, so you can see that groove in the middle of that dinoflagellate on the right-hand image at the top. That's where one flagella hangs out at, basically wraps around the dinoflagellate, and then you have the second one hanging off of it, and that second one is what they use to move around with. Dinoflagellates have armored plates made from cellulose, so that is a type of carbohydrate that protects them, and they tend to have a lot of spines, pores, and other ornaments to help them stay afloat. So those pores allow the exchange of water and nutrients within their body. Dinoflagellates can be harmful not only to humans but also to the organisms within the ocean. Some species of these dinoflagellates are known to hunt prey, and they have what they call an eye, but it's not really an eye that you would think of like how we have eyes and we can see. But they look they look for changes in light, so it would be a light sensitive eye, and it helps them hunt for prey. And some other ones can actually bioluminesce. So that image on the bottom right hand corner there are dinoflagellates who are bioluminescing in the evening, and they're creating their own light. And let's see, dinoflagellates are also responsible for red tides and ciguatera within fish. So be very careful about eating reef fish, in particular barracuda and other reef fish like manini and whatnot. Those fishes tend to have this dinoflagellate within their fleshy meat. And if you eat fish that have been poisoned with ciguatera, you will get very, very sick. And then there's other dinoflagellates that tend to be parasitic, so they will create sores or create diseases within shellfish and humans. And you can see the top right hand image over there. You can see those sores on those fish there. That is caused by a parasitic dinoflagellate. Alright, and then you have others that are known as zooxanthellae. Zooxanthellae are dinoflagellates that are found within corals, giant clams, and sea anemones. And these guys have a mutualistic relationship with the other organism. So on the right hand image there, you can see the zooxanthellae cells, which are purple, and they are found within the skin of the coral polyp. So the main job for the zooxanthellae is they fix carbon dioxide that is released from the polyp, and they convert it into oxygen and nutrients using photosynthesis. And they also help in building the coral skeleton. So they, the zooxanthellae, it's an advantageous partnership because the zooxanthellae, you get carbon dioxide from the polyp, and the polyp then gets oxygen and nutrients. And so both of them are getting exactly what they need in order to survive. All right. And then you have protozoans. Protozoans are animal-like eukaryotic cells. They can be heterotrophic. Most of them are heterotrophic, so they need to eat. But some of them can photosynthesize, but that's not very common. And so you can see the image below there. You can have some different types. You can have rhizopoda, ciliate. You can have ciliates, basically the ciliata, whatever that's called. Those are ciliates. Flagellata, sporosa, whatever. Anyways, you have parameciums, euglenas, and amoebas. Yes, amoeba is actually a category in the protozoans. So, yeah, those are fun. And then you have foraminiferas, or forams for short, because I hate saying this word. These particular protozoans they have a calcium carbonate shell. So these guys don't photosynthesize. They have to eat their own food. And if you look on the image on the right hand side there on the bottom, you can see these spine-like things coming up from the top, which are called pseudopodia. So essentially pseudopodia just means like fake feet, if you really break down the parts of the word there. But what they do is they put out these spines, and they're sticky now, and they capture food, and they bring the food back into the cell itself, and that's how they eat. All right, but essentially that pseudopodia are extensions of the cytoplasm. And these particular shells, you can look at the top right image there. They grow with the cell. So unlike the dinoflagellates that you saw where the shells come apart, these particular organisms continue to grow their shell around them, so they can get pretty large, as you can see by the individual in the middle there. And these forams can either be planktonic or benthic, so that means they can either live within the water column near the surface of the water, or they can live on the bottom of the ocean. These particular guys, because they are made of calcium carbonate, or their shell is made out of calcium carbonate, they use the calcium carbonate shell to infer what global temperatures were like in the past. And so, for instance, if they see that there's a lot of these what's known as microfossils within an area, then they know that conditions or temperatures were favorable because there were a lot of them. And if they don't see a lot of them within an area, then they within a given time period, I should say, then you can tell that there wasn't the conditions were not favorable, so you didn't see a lot of these guys around. Also, too, the calcium carbonate test, when it's formed, it stores oxygen within its chest. And so when they find these shells, they test the chemical signature of the oxygen within the shell. And if it's a particular isotope, then they know that the temperature was warmer or colder than it normally would have been. So when you find a lot of oxygen 18, then you can tell that the temperature on Earth in Earth at that time was colder, because oxygen 18 tends to like colder temperatures. And when you find a lot of oxygen 16, that's when you have warmer temperatures. Then you have radiolarians. These are protists. They have to eat their food too. You can see they have all different shapes and sizes. They have all different types of spines. Some of them are round, some of them are UFO shaped. I really like the UFO shaped ones, especially the one on the bottom right hand corner there, with the 3 legs coming out. That's pretty cool. Anyways, these particular guys make their shells out of silica, and they have tons of spines, and those spines are used for keeping themselves buoyant and up in the water column near the surface of the water. Their shells, when they fall down to the bottom of the ocean, will create what's known as siliceous ooze. So it's basically just a bunch of shells, right? They're shells, and it's not really ooze like gooey ooze. It's just I don't know why they call it an ooze, but it's basically just meaning that this particular sediment has a greater proportion of radiolarian shells within it. All right, and then we have ciliates. Ciliates are pretty interesting because they use their cilia to move around. So those fine little hairs that you see on the outside of that paramecium there, and that's what they use to move around and hunt their prey. They don't have a flagella, they only have cilia on the outside. You can find ciliates within freshwater or the ocean. They're in all bodies of water, basically. You can find some that live within organisms, like the inside of the intestines of a sea urchin, or you can find them in the gills of clams, or you can find them within the skin of fish. So these particular ones, I probably would not call them mutualistic, like the zooxanthellae and the coral. This is probably more of a one-sided relationship, where the ciliate living within the intestines of a sea urchin are taking advantage of all the nutrients within the intestines of the sea urchin, but they're not giving anything back to the sea urchin. So it's definitely not a mutual, both parties are not benefiting from this relationship. And ciliates are a very important source of dissolved inorganic matter. So a lot of the nutrients that go back into the ocean are from these guys right here. And lastly, we have fungi. You can create a long filament of fungi known as a hyphae. These particular organisms lack chlorophyll, so they do not photosynthesize. They do need to get their food somehow, because they are definitely not heterotrophic. And what they do is they absorb nutrients through their cell walls, which are made out of chitin, and they decompose detritus, which is dead matter, basically. So you can see on the top right hand image there, that orange hue on those rocks, those are that is fungi, a marine fungi. And the bottom right hand image there is a close-up image of what the fungi looks like. You can see it has spores, so those orange round dots that you see within the image are its spores. And then you can see the long chains, the long filament known as hyphae within that image. And when you combine algae and fungi, you get a lichen, much like how you get a lichen on land. Marine lichens are nitrogen fixers. So they do fix nitrogen, turn it into ammonia, then turn it into nitrate which then gets turned into nitrite. All right, so that was chapter 5. So stay tuned. We will talk about chapter 6, which I believe is on invertebrates, but don't quote me on that. I am a little tired right now, so I will let you go. I hope you're not listening to this lecture too late at night, like how I am right now. So I will see you in class later on.